So one of the things that we're going to be tracking as we're learning physiology in this course is the actual mechanism of changing these various regulated variables in our body, as well as other types of signals. So to understand how homeostasis works and to understand how these feedback loops work within specific organ systems, we're going to want to think about a couple key components. So we have key structures that allow us to maintain homeostasis. This is a typical pathway for a negative feedback loop. So what we have happen in our typical pathway to maintain a homeostatically regulated variable, we first have, I want a pointer here, okay. So we start out with some sort of change in the body. So here, we're looking at a change in blood pressure due to a change in position. So when you're lying down, your blood is all pooled here along the backside. When you stand up, suddenly your blood starts to pull down. It gets pulled down by gravity. And this is why sometimes you'll experience kind of a head rush when you stand up. Because when you stand up, your blood pressure actually falls due to this change in the position of your body. We call this a stimulus. And so this stimulus, this change, would be registered by a receptor. So our receptor here is going to sense this change in blood pressure, which is actually our regulated variable. So your position isn't regulated, right? You can sit down, you can lie down, you can stand up, you can do jumping jacks. Doesn't matter, but your blood pressure, we want to keep the same. So blood pressure falls, it's regulated, so this is going to be a stimulus for change when it's noted by the receptor. So information about this change in blood pressure makes its way to the receptor, and then that receptor will often signal to an integrating center. So some things are controlled locally, but a lot of things are controlled by having a lot of organ systems potentially working together, a lot of things your brain needs to know about. So the sensor, the sensor sends the signal to your integrating center, which is going to sort of plan out the response. So then we'll have another signal going from your integrating center to the effectors. So we'll have a lot of different organs that can be effector organs. One of them is your heart. So here we see we have a signal going to the heart, which is our effector organ. And what it does is it increases the heart rate in order to get blood pumping faster to maintain that blood pressure in this new position. So this is our actual negative feedback response. So we had to go through this whole pathway in order to change the blood pressure. So as this heart rate increases, the blood pressure increases. That's the opposite type of change than we started with. So we started with blood pressure falling, we end with blood pressure rising. So this is our negative feedback response. And we'll see similar pathways throughout this course. So we want to be familiar with this basic structure. Okay. So here we have a couple of examples of these different sort of steps of this pathway. So remember, I'm going to flip back to the last slide for a second. So we're going from a receptor, sending a signal to an integrating center, sending a signal to an effector, and then we're going to have a change at the end, a signal that's in the opposite direction than our original signal. So our receptors that are going to detect stimuli include thermoreceptors, sensing heat, chemoreceptors, sensing concentrations of specific chemicals in the body, baroreceptors, sensing pressure. We'll have different types of integrating centers. Most of them are in the brain. Some of them might be in your spinal column. These are where we're orchestrating the appropriate response. We'll have different types of signals. We'll have signals coming from the receptor to the integrating center. We'll have signals going from the integrating center out to the effector organs, and we'll talk about what those signals look like, and what nerves look like, what neurons look like in our next unit. And we'll then move into effector organs later in the course. So some effector organs are muscles, which we'll get to. We'll also see glands, different parts of your endocrine system responsible for hormones. Those will also potentially participate in these negative feedback loops controlling our regulated variables. Okay. So here's a diagram of what that pathway looks like. This one doesn't have a specific example written out, right? This is just the general pathway. So we start here, number one, and we start with our variable. And there's some change in it. So here we can see that there's an upward arrow by the x. So we have this variable x rising. So variable x is rising. It's some regulated variable. Here you can see this diagram at the top is actually synced up with this diagram below it, right? So here we have x rising, also shown here by this pink line. And it's going to activate a sensor. So here we see this box in purple, x rose, the sensor sensed it. So that would happen somewhere here along this rise. Then we're going to send that signal to the integrating center. The integrating center is going to plan out a response. Once it knows what that response is, it's going to send out a signal to the effector organ. When the effector organ is activated, that's when we're going to see the corresponding decrease in our regulated variable. So we'll see we had a rise, our body sensed it, sent it to the integrating center planned a response, effector organ affected that response, brought us back to our set point. And this will happen within a normal range that your body is able to control. So your temperature, you have a set point, but there's also a range of temperatures that your body is well equipped to handle using these negative feedback loops to return to the set point. However, in some cases, we exceed this normal range and we'll get into areas of the graph where it's difficult for your body to actually pull our regulated variables back down to their appropriate set points. So this is why, for example, really high fevers are a problem, is it can actually mess with our thermal regulation. At certain extreme levels, this becomes a lot more difficult for the body. Okay. So in a negative feedback loop, we're talking about a move in the opposite direction from the change. Our error signal is decreasing, so this distance from the set point is decreasing once our effector is activated. It's decreasing because we're actually having an effect. It gets us back to the set point. And this is always going on. So you're always having slight perturbations in these regulated variables. Your body is constantly making very small adjustments. So here's our example from the beginning, body temperature. So there are sensors in your brain, actually, that detect a deviation from the set point of 37 degrees Celsius. So our sensor is in the brain, sensing a change. And we also have an integrating center in the brain in another part of the brain. So our sensor in the brain sends a signal to the part of the brain that's responsible for creating a response. And then that part of the brain sends out a signal to the effectors, which in this case are largely sweat glands, which produce water on the skin, allow it to evaporate to cool the body. And once the body is cool, the sensors again tell the integrating center that we're cool enough now. So it's sending a signal again to the integrating center and telling it to stop turning on these sweat glands. So we'd see sort of a fluctuation, right? So as we're sweating really hard, we pull that temperature back down because sweat is evaporating off our skin. And we actually may bypass our set point, right? We go a little bit past it. We're still sweating at this point. Oh no, we get too cold. So then the sensors send a signal to the integrating center, bring us back up. So we can expect to see often these continuing curves, these fluctuations. Homeostasis often uses multiple effectors that move conditions in the body in opposite directions. So we have this dynamic constancy. So in this case, when we're talking about opposing effectors, we're looking at this fact that you can sweat if you're hot, or you can shiver if you're cold. So we have these two mechanisms to change your body temperature, and they pull or push body temperature in different directions. And as you oscillate from one to the other, you can end up with this sort of sine curve, which we refer to as our dynamic constancy. So if you averaged this all out, even though we're going a little bit above 37, then a little below 37, you'd average out to the set point. We never deviate from it too far. Other things that are regulated this way include blood glucose levels, so sugars in the blood, allowing you to remain metabolically active, allowing your cells to get energy. Blood pH is another good one. So if we're talking about blood glucose here, we see the same type of loop, right? It's a negative feedback loop. So we'll see a lot of diagrams like this throughout this course. So keep an eye on these dotted arrows with a minus sign. So this is a negative feedback loop. This is telling you that we're going to be inhibiting this at the end of the process. So as we go through this process of maintaining our normal blood glucose level, sometimes we're going to do something that changes blood glucose, right? You eat food, you eat a bunch of crackers, you eat a donut, your blood glucose goes up. Okay. So then we have a sensor that senses levels of blood glucose. The sensors send signals to the pancreatic islets of Langerhans, which produce insulin. Insulin allows your cells to pull in more glucose. And because they're pulling in glucose from the blood, the glucose remaining in the blood goes down. And now that the fact that the blood glucose goes down, we've used this negative feedback loop to inhibit this whole pathway. So this negative feedback loop, we've pulled blood glucose back down. So this whole process gets kind of shut off. We're not going to send these signals. We're not going to have these effects anymore once our blood glucose is down. And it works the same way if we're fasting. We have a process that allows us to do this in the reverse. So if our blood glucose drops, we send signals to the pancreas, which actually decrease insulin and increase something called glucagon. And glucagon increases glucose secretion into the blood by the liver. So we're putting more glucose in the bloodstream, and blood glucose goes back up. And then we shut off this whole pathway now that blood glucose has returned to its set point. So we have another minus sign over here. So whenever you see a diagram like this in your textbook, pay some attention to it, because we use these diagrams to basically summarize really important concepts. And I often find that they're a little easier to interpret than reading pages and pages about this in your textbook. But to understand them, you really have to think critically. Okay. So we do also have positive feedback. So negative feedback, our end result, inhibited the process that we're talking about. So it turned off that loop. In positive feedback, we think about it like a runaway train or like a snowball rolling down a hill. In positive feedback, the end product of our process actually stimulates that process to happen more and more and more. So we think about it as amplifying a change. So we do eventually have to turn off positive feedback loops. We don't want a runaway signal forever and forever and forever, but we'll see positive feedback when we're talking about a lot of different types of signaling, we'll see that positive feedback has an important role, for example, in blood clots and in various parts of the menstrual cycle. Okay, so one example of positive feedback in terms of blood clots, right? As your blood starts to form a clot, where a blood vessel is damaged, you don't want your body to notice that it's making a clot and immediately get rid of the clot. So as we're forming this blood clot, for a little while, that process, that end plot point, that fact that a clot is forming along the wall of the blood vessel, actually attracts more and more and more and more platelets in order to fully form the clot. But if we kept forming the clot forever, we'd eventually block the blood vessel, which is not ideal. So once the damage is fixed, we'll have a secondary process that ends clotting via negative feedback. So clotting starts out with a positive feedback mechanism that's amplifying a signal, and then it'll end with a negative feedback loop. Uterine contractions in childbirth are also regulated by positive feedback loops. Like contractions get stronger and stronger for a while until the baby is born, when we then have a negative feedback loop to turn off that contraction. So here we see that estrogen secretion by the ovaries is also under positive feedback control. So your pituitary gland, which is a gland in your brain, secretes something called luteinizing hormone. So luteinizing hormone gets sent to ovaries in women. And in the ovaries, it increases estrogen secretion. This is a positive feedback loop. So that estrogen goes back to the brain, gets to the pituitary gland, and the pituitary gland secretes more and more luteinizing hormone. And this is actually what spurs ovulation. So this surge for a little while of luteinizing hormone. At some point, we'll then have a negative feedback returning the levels of this hormone back to normal. But for a little while, this process is going, and it's really important to how your reproductive system is functioning. So when we're talking about regulating our regulated variables, what we've been talking about right now largely is extrinsic regulation. So when we're talking about a stimulus that is sensed and then sends a signal to an integrating center in the brain, we're talking about an extrinsically regulated variable because we have to go man a response somewhere away from the place that the change is actually taking place. We will also see that in some cases we have intrinsic regulation of our regulated variables. So in intrinsically regulated processes or intrinsically regulated changes, we'll see that the local cells, those cells within the organ itself that's sensing the change, send a signal to neighboring cells to respond appropriately. So we don't have to go all the way up to the brain and back down to another organ in every case. Sometimes the response will be right there where we sensed a change. So we will see both types of processes as we go through this course. Okay. We're going to take a short break while you pull out your notes on biomolecules and enzymes, which is our next topic. And if people have questions at this point about homeostasis, now's your time. You should feel relatively comfortable with biomolecules from what you learned in your prerequisites, but it's important that we understand some of the key components of biomolecules and how they function to be able to understand how your body produces them, uses them, and breaks them down, which are all processes we're going to talk about as we go through our different organ systems. Okay. So our objectives for right now are to understand what a biomolecule is, which means we want to know what the 4 groups of biomolecules are, how they're different from each other, both in terms of how they look, if we're just looking at a chemical diagram of a biomolecule, but really importantly, we want to know about how they're different functionally. So we want to know some of their chemical properties. Specifically, we want to really key into how they're going to respond to water. Water and ions, I would say, we really want to be tracking when we're thinking about biomolecules, because those are the types of interactions we're going to be thinking about as we look at specific cell types. Secondly, we want to understand the role of enzymes, so specialized types of proteins, and how they work in chemical reactions, because we're going to see a lot of different enzymes in this course that are responsible for producing various substances, for regulating various processes in the physiology of our body. So we're going to start with our groups of biomolecules. So this part should be a review. Okay. So when we say biomolecules, you may also have heard them referred to as macromolecules. Same thing. So we're thinking about molecules that are synthesized by living things. There are a lot of different chemicals in the world, but living things make a couple key types. They're all based on carbon. So if we're talking about a biomolecule, it will contain carbon. You will not see a biomolecule that does not contain carbon. It'll have a couple other components, some really important ones. I would actually reorder this list. So hydrogen is going to be really important. So we're going to see carbon and we're going to see hydrogen. We're also going to see variable amounts of oxygen. We're also going to want to key into whenever we see nitrogen, and also things like phosphorus. Occasionally we'll see sulfur, but these are going to be our key components of biomolecules. We have 4 basic types of biomolecules, which can be distinguished based on their chemical formulas, based on their structure, and they have different functions within the body. So our 4 basic types of biomolecules are carbohydrates, we got our lipids, we got our proteins, and we got our nucleic acids. We're going to cover these each in order. So we're going to start with carbohydrates. So we're talking about carbohydrates, we're talking about things in the real world like bread, pasta, sugars. In the body, we're also talking about structures that store carbohydrates. So glycogen is one we'll talk a lot about when we talk about the liver and metabolism and how we can store glucose. So glycogen is a really important carbohydrate biomolecule when we're talking about physiology. So carbohydrates, we'll see, always contain carbon, hydrogen, and oxygen. So those are the 3 things that you should see in a carbohydrate. If you see nitrogen, you're not looking at a carbohydrate. We'll also see that they're usually in these sort of ring type structures. So we can see that we have a mostly carbon ring containing an oxygen molecule surrounded by hydrogens and hydroxyl groups. Okay. We have 3 types of carbohydrates that we will name kind of specifically. So groupings of carbohydrates, we can group on the number of rings they have. So if we see 1 ring, we're talking about a monosaccharide. If we see 2 rings, we're talking about a disaccharide. If we see lots and lots of rings, we're talking about a polysaccharide. This will be a little bit important when we eventually start to talk about digestion, because we'll see that these different sizes of carbohydrates will be broken down in different ways. So the way we make a larger carbohydrate is by something called a condensation reaction. So when your body stores carbohydrates, when it stores sugars, it doesn't store them all as just tons of glucose in your blood. We talked about just now that glucose is a regulated variable, blood glucose. So you can't just shove a ton of glucose in the blood after you eat. So it needs to go somewhere it needs to get stored. So in order to store sugars, we're going to link together our monosaccharides using covalent bonds. We're going to link them together into larger polysaccharides. But here on the slides, we have an example of the first step, which is to make a disaccharide. So when we link together two monosaccharides, we link them together right here. So we take these two hydroxyl groups, we join them together, and when we join them together, when we form this covalent bond to oxygen, and then the covalent bond over here to the second monosaccharide, we have to get rid of some stuff. So you can see that here, when we have two monosaccharides, we have two hydroxyl groups, so one on each of these monosaccharide rings. But when we move into this disaccharide, we can see there's just this oxygen here. And so we're missing a hydrogen here, and we're missing our second oxygen. So when we link together carbohydrates, we have a condensation reaction that releases water. So that's where those other atoms went. They became water and they floated off. We can also do the reverse. So we're also going to want to be able to break down any carbohydrates that we've stored in the body in order to use them when we're not eating. So you go on a run, that's when you're going to really want a bunch of blood glucose in order to rapidly fuel your metabolic processes. So we're going to want to be able to split up our carbohydrates, and we're going to do this through hydrolysis. So we're going to split together larger saccharides. So in this example, a disaccharide, but also polysaccharides. We're going to split them into smaller components by adding water. So we have to add back in those atoms in the form of water, this hydrogen, this other hydrogen, and a second oxygen in order to get back to this form, because otherwise we don't have enough atoms. We don't have them in the right amounts. Okay. So if we add water to a polysaccharide to split it into smaller carbohydrates, we're doing a hydrolysis reaction, we're going to need to put in water. Questions on carbohydrates before we move on to lipids? Okay, not hearing anything. If anybody sees something in the chat that I haven't addressed when I'm sharing my screen with you like this, it's very difficult to see the chat. So if someone types something out in the chat that you want asked out loud, I'd appreciate it if you help your classmates out and read those out for me. Okay, we're gonna move on to lipids. So lipids are different than carbohydrates, partially because we're not going to see oxygen. So lipids can sometimes be really, really simple chemically molecules. So we're going to have just hydrogen and carbon, and we'll sometimes see other groups attached at the ends of lipids, but they don't form the main part of our lipid molecule. And because our lipids are composed only of hydrogen and carbon have similar electronegativity, which means that a bond between a carbon atom and a hydrogen atom is nonpolar. And because lipids are almost entirely carbon and hydrogen, this means that our lipids are nonpolar. And polarity is really important when we're talking about physiology, because your body is mostly water. Nonpolar molecules don't interact with water. They're repelled by water, which means that they're hydrophobic or water-fearing, which means that if you have a lipid in your body, it's not going to mix well with your blood plasma. Okay, so we're going to see that when we get to digestion, for example, we'll have really different mechanisms for breaking down lipids, because they're going to kind of clump up together in your blood sometimes because they're repelled by that water in your blood, in your plasma. Okay. We do have different kinds of lipids though, so that's Not always true for every single kind of lipid. That's a generalization. What's not a generalization is that we are always going to see these carbon and hydrogen atoms. Okay, so we have 5 basic classes of lipids. We have triglycerides, we have ketones, phospholipids, eicosanoids, and steroids. That word always trips me out. Okay, so we're grouping these together because they're mainly carbon and hydrogen, and they're going to have at least nonpolar parts. We'll see, for example, in phospholipids, it's going to be really important that part of that molecule is polar, but we also have a nonpolar part because it's a lipid. Your most textbook example of a lipid is a triglyceride, so that's the first one. So our first class of lipid are triglyceride. It's made up of glycerol and 3 fatty acids. So glycerol here is the backbone of this molecule. So this sort of flagpole part. It's a 3-carbon alcohol. And it joins together our fatty acids. These fatty acids are really what make triglycerides a lipid. So you can see that the fatty acids are just carbon, hydrogen, hydrogen, carbon, hydrogen, hydrogen, carbon, hydrogen, hydrogen, kind of forever. So we have these long carbon-hydrogen chains that we call fatty acids. 3 of them are bound together to form our triglyceride, right? We have 3 of them. Our fatty acids don't change in terms of the atoms they contain, right? Like they're always this carbon-hydrogen chain, but the shape of them can be different. So sometimes we will see double bonds in this chain, and that's going to change the shape of the chain, which is why we'll see that different triglycerides have different functions in the body, behave differently. So we have saturated and unsaturated fatty acids. A saturated fatty acid, when we say saturated, right, like in normal English, that means like kind of full of something. So what these fatty acid chains are full of is hydrogen. So we have the maximum amount of hydrogen that fatty acid chain can hold. They're saturated with hydrogen. But if we put a double bond here, if we have a double bond in our fatty acid chain, we are now going to have not so many hydrogens because we've taken up a bonding space, right? So if we see a double bond, we're talking about an unsaturated fatty acid, which will either have the remaining hydrogens on the same side or on opposite sides. So we have our trans fatty acids and we have our cis fatty acids. Cis on the same side, trans on opposite sides. And we will pick up here on Wednesday.